while I pass out the exams. Okay, so our membranes, right? So again, I think a lot of the things I'm going to say here is nothing surprising because you've probably seen all this before in multiple biology courses, probably in high school up through freshman biology. Um, the way the lipid bilayer is structured is different than what the micelle is structured. From a physical perspective, the idea is the same, right? We are grouping together the non-polar regions and the polar regions, okay? Because of the nature of now our phospholipid and the fact that we have two carbon chains attached to a polar group and not just a single carbon chain, that actually is really, really important for the reason that chemically speaking, physically speaking, that the bilayer forms and not the micelle. Okay, but at the end of the day, what really matters is that these phospholipids orient themselves such that we have this bilayer with the interior being all hydrophobic and the exterior being where our polar head groups are. The other thing to note here is that in this cartoon diagram, it shows two chains attached to the polar head group. And it shows two chains because on our glycerol backbone, there are actually two carbon chains. What it doesn't show well is the fact that these are not lined up perfectly. They're not lined up perfectly because one of these carbons is always unsaturated. So in reality, they don't line up perfectly like this. One of them is always kinked, so things are a little bit more fluid, right? The idea is that one of them gives a lot of space for things to move around. Okay? Because membranes are fluid, because of that unsaturated fatty acid, things are able to move. And proteins can move. We're going to see there's lots of proteins in bilayers, but lipids themselves, the fatty acids themselves, can actually move around as well. There's two types of motion. One is called lateral, and this idea is pretty simple, is that just things are dynamic enough that one fatty acid, one phosphatidyl, whatever it might be, can move around as long as it doesn't flip, right? It just moves around. This is in equilibrium. There's no preference for where it's positioned, so it can move. The other type of movement is called transverse. And the idea here is you have a flipping, right? So one of the polar head groups reorients itself so that it's positioned on the other side of the bilayer. And as you might imagine, this is a lot harder to do because you're taking something that's hydrophilic, oftentimes charged, pushing it through this hydrophobic region where it really doesn't want to be so that it can reorient itself pointing down. Okay? Biologically speaking, this happens, and when it does, it's really important. It's usually like a signal for something. But kinetically, it's hard, right? Free energy-wise, there's no difference. This polar head group is situated here. It's interacting with water, a polar environment. It's happy. Same thing once it reaches its final place. So thermodynamically, it's no big deal. Kinetically, though, there's this massive activation energy to try to force this hydrophilic thing through this very nonpolar region. So this tends to be facilitated by an enzyme of some sort. Okay? This idea of fluidity is important, right? And cells are very good at controlling how fluid membranes are, and they do so using one of our other classes of lipids, and that's cholesterol, right? The role of cholesterol in our cells is to control the fluidity of a membrane. So the idea here is that where this unsaturated fatty acid is positioned, it kind of creates a gap in our bilayer. And cholesterol can orient itself in there, filling that space so that we maximize our London dispersion forces and that rigidifies the membrane. So the amount of cholesterol that's embedded in our bilayer will control how fluid or how dynamic the bilayer is. That's the role of cholesterol. And if you look at the structure of cholesterol, there's that single hydroxide group. So the only way it can orient itself is with that single hydroxyl group positioned at the polar head group, our four-ring system positioned where that gap is, and then this carbon tail then sits further down into the bilayer. Okay, so then you can kind of take this idea and think about the composition of a bilayer, right? So if you look at the protein to lipid to carbohydrate ratio, these bilayers specialize. And we're not going to go through everything here, but what I do think is interesting and you should kind of like take note of is the difference between bacterial membranes and eukaryotic plasma membranes. The massive difference here is in bacteria, you have a lot more protein content than lipid content. In fact, it's most similar to what we see in the mitochondrial inner membrane. So remembering that prokaryotes don't have organelles, right? They don't have a mitochondria, they don't have a nucleus. What do you think might have to happen in the plasma membrane for bacteria that otherwise would typically happen in the mitochondrial inner membrane that requires lots of proteins? Hmm? Yeah. Yeah, so oxidative phosphorylation, right? So like all of the four protein complexes that are important for the electron transport chain, typically we think of in the mitochondria, but in bacteria we don't have that. Right? They don't have a mitochondria, so all that happens in the plasma membrane. So the reason that there's so much more protein content in the plasma membrane for bacteria is because that's where they make their ATP. That's where they create their proton gradient to drive ATP synthesis. Okay. Um, so when we introduced the idea of like, you know, protein structure and we thought about how amino acids are arranged, we kind of introduced this idea of membrane proteins. We introduced the idea of like peripheral membrane proteins or integral membrane proteins. Integral membrane proteins are those that orient themselves completely in the membrane and span the entire length of the membrane. Okay, so we're not going to go into the structure a lot, but suffice to say that there can be multiple ways of doing this, right? We can accomplish this in many ways. A single alpha helix, multiple helices, or this really cool, it's called a beta barrel. And these tend to form porins, which have big pores the molecules can go through. Like water, the water channel in membranes happens through these beta barrels. And it just kind of like this opens up this big old pore that water can easily go in and out of. You might be able to very easily think about what properties the amino acids in the core of the membrane have to be versus on the outside of the membrane. Peripheral membrane proteins are those that are embedded in the membrane but don't span the entire region, right? So the peripheral membrane proteins can be in lots of different ways, right? So here we see an alpha helix that's embedded and interacting on one face of the membrane but doesn't make its way all the way through. Here we see, so this protein is attached to a fatty acid, and that fatty acid anchors it into the membrane. You can have an integral membrane protein that interacts with a peripheral membrane protein that allows this protein that otherwise wouldn't associate with the membrane to have a reasonably high affinity so it can interact with the membrane. Okay, lots of different ways that we see these proteins interacting with the membrane. And I think this is the last thing that I have on here. We'll leave with this idea that within the bilayer, right, these cells have evolved in a way where lots and lots and lots of proteins cluster together in what we call lipid rafts. They are sections within the lipid bilayer where lots of proteins are associating with each other. Okay, and the reason for this is efficiency. So if we have a surface area that's the table, right? If this is like the surface of a cell, and we know that there are 15 different proteins that all need to work together to accomplish the same goal, ATP synthesis, or whatever it might be, it wouldn't make sense for those proteins to be positioned randomly throughout our surface. Instead, they position themselves so that they cluster together, they have an affinity for each other, they cluster themselves together in a really similar space. And that space is called a lipid raft. It's regions of the lipid bilayer with massive protein density, and all the proteins there work together to accomplish similar things. Okay? Oh, there's more. Okay, so really quick here. So the way that we can move things across a membrane um, is, so we have multiple types of transport, right? Some molecules are able to just randomly diffuse, just spontaneously diffuse across the membrane. Oxygen, for example, right? The oxygen we breathe, the hemoglobin takes to our tissues, there's nothing. So once it gets to our cells, oxygen does not have any type of protein that moves it into our mitochondria. It just diffuses across the membrane because it's hydrophobic. It's got enough properties that allow it to dissolve in water, but it's hydrophobic, so it can spontaneously diffuse across the membrane according to just a concentration gradient. Most molecules don't have that ability, right? So here we have something called a facilitated uniporter. The idea here is the molecule follows a concentration gradient to get across the membrane. So here you have a high concentration, on the other side a low concentration. So energetically speaking, it's spontaneous to move something across the membrane. But that molecule can't do it by itself. It's not oxygen. It's not nonpolar. So we have these proteins that are able to do it. They're proteins that have evolved to specifically recognize this molecule and help it pass across. These are facilitated diffusion proteins. Okay, they're also, so here's another example of it, right? So glucose, really important to get sugar inside of a cell. The way that it works is glucose, so this glucose transporter recognizes glucose. When glucose binds, it triggers a structural change that there's a conformational change in the protein that then releases it to the inside. And then once it's released, it then returns to the structure where the binding site is exposed to the outside. And the other type is active transport, right? This is a type of transport that it goes against a concentration gradient. Okay? And as the name implies, it's active, which means we have to actively provide energy to do this. And one example of this is consuming ATP energy to move ions across a concentration gradient, right? So in this case, we're moving sodium out and we're moving potassium in. But to do so, we need to consume ATP energy. Okay? And I do believe that is it. Good enough. Good enough. Okay. All right, any questions? All right, so for the last 12 minutes, feel free to go ahead and work on your